For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 2, Episode 60 of Surfing the NASH Tsunami. Our preview of NASH-TAG 2022 starts now.
This week on Surfing the NASH Tsunami.
I'm very excited to say we brought back the oral presentations. We didn't have any oral abstract presentations last year. Everything was poster. We have had the best crop of abstract submissions we've ever had at NASH-TAG. I've reviewed abstracts for a long time for all the major congresses, and I can tell you that many of these that were submitted would be taken as orals at AASLD or EASL, and a lot of them are novel.
This is really a great platform for those amazing research networks that have been formed on non-invasive biomarkers at LITMUS presented by Quentin Onsley and NIMBLE by Rune Sanyal, and then of course the Emerging Cirrhosis Network. Those are investigator-driven or academia-driven consortia where we think the biggest unmet need is, and they have really just generated amazing amounts of data.
I'm interested in the why NASH-related HCC is resistant to treatment, because it begs that we have to diagnose earlier, we have to start screening earlier, particularly if we have a form of cancer that is really resistant. It really pushes that paradigm further forward for the screening of type 2 diabetes.
The last session has the promise to be the best session anybody's ever seen anywhere in fatty liver disease, if in fact it becomes the kind of exchange it's capable of being. I just find it so exceptional you guys have pulled that together. It's just going to be such a remarkable event to watch.
Thank you. I think it's a testament to the fact that every one of these stakeholders feels the exact same way. From the FDA to sponsors to key opinion leaders and everybody in between. Everybody wants to find a way to get the right drugs to our patients as quickly as we possibly can and as safely as we can. And so we all have the end goal the same.
In January, a group of several hundred leading fatty liver disease stakeholders will gather in person at Deer Valley, Utah, or virtually from around the globe for NASH-TAG, a meeting for drug developers, researchers, and investors to learn the latest research and discuss the most critical issues in NAFLD and NASH today. Join hepatology researcher and key opinion leader, Dr. Stephen Harrison, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and this week's guest, hepatology researcher and key opinion leader Professor Jörn Schattenberg, as they preview the NASH-TAG agenda, this week on Surfing the NASH Tsunami.
So, welcome to the final lap of Surfing the NASH Tsunami for 2022. NASH-TAG is my favorite event on the Fatty NASH calendar. I know I'm not alone in that. Now, I am alone in that NASH-TAG is the first place I encountered the NASH community, so that's a little different. But many folks share my belief that this meeting provides a unique opportunity for deep, thoughtful, and informal conversation among friends and colleagues. One of the highest compliments this podcast has ever received came when someone told me, and I quote, "If NASH-TAG side conversations were compiled in a podcast, this is what it would sound like." So I love that comment. We'll try to see if we can live up to it today. This is a really simple session. Stephen, who's one of the NASH-TAG co-chairs, will be walking us through session by session, and Jörn, who's a steering committee member, will be providing feedback, and then Louise and I will ask questions. In previous years, we've been successful in getting a different co-chair to join us, but Rohit is in India, Michael's on his honeymoon, and Vlad is getting ready for a big trip to New York. Stephen will have to, as he's perfectly capable of doing, play the role of two people, although we're not going to ask him to use two different voices to do it. So with that, let's get started. Usual icebreaker, one good thing, personal, professional, about the last week. Brave one, go first, Stephen. I hear you can talk forever on this.
Well, it's been an exciting week. On a personal note, if you've listened to the podcast for any length of time, you know that I am a huge Ole Miss Rebel football fan. We finished the year number 8 in the country, and because Alabama beat Georgia, we are now going to the Sugar Bowl. And the Sugar Bowl is a huge honor to be able to play in. We're going to be playing Baylor on January 1st, so exciting times there. And then you can call this professional or you can call this personal, but I just got back from Merida, Mexico last night, where I attended the wedding of Mary Ranella and Michael Charlton, and it was amazing. It was an event that I've known both of them for 2 decades, and I don't think I've ever seen them happier. That is something that will resonate with me for a long time, seeing their happiness and their joy with each other. And just to be a part of that and the whole experience was something I'll remember forever and cherish, and I'm so happy for both of them. Next time you see see either one of them, congratulate them, and they're likely to have a smile on their face from now until forever. So it was a great time.
Fantastic, Stephen. Who wants to go next?
Oh, I'll jump in next. Starting to wind down for Christmas personally, but trying to catch up on a nutrition course that I'm in the middle of, to be a nutritionist, basically to aid more information that I can give. It's giving me some nice thinking processes towards micronutrient and vitamin deficiencies and how they could affect both the placebo groups and also the responding groups. Enjoying doing that.
You're making fun of my Lay's today.
I was making fun of you eating crisps, as it takes 4 kilos of potatoes to get the equivalent fat content of a packet of crisps. Just think about that next time you look at 4 kilos of potatoes.
Yeah, so Stephen, how many grams exactly is in that package? It should tell you on the package.
Let's see. Total fat grams are 14.
No, calorie. Total grams of the food content.
Well, the calories are 230. Fat grams are 14. Carbs are 24. 3 grams of protein. Come on.
So that's a multiplier of 40 to 1 or something like that?
I'm eating it for the protein.
Every gram of fat has 9 kilocalories, so
it's comfort food, all right?
We'll turn this into a nutrition webcast.
You're promoting NASH-TAG in the opposite direction.
I have 2% liver fat on my PDFF, and my CT1 is 600. So I'm allowed an occasional small, not big, bag of chips.
I thought that until somebody did my cholesterols recently, and they were way too high, and yet I have no liver fat either.
So you aren't follow that.
Yeah, it's difficult, right? Talking about food, it's Nikolaus Day here in Europe. So my daughters were prepping their boots that were positioned in front of the house last night. And this morning, having them come down, they were all filled with chocolate and cookies. I guess that's a tradition over here. So that was joy to see them happy on that occasion. Professionally, over the weekend, I had the opportunity to review the liver ASLD abstracts that were submitted to the American Gastroenterologists Association AGA meeting in April, so it's excitement there. And actually just got off the phone for the UEG, which is the European Gastroenterologist equivalent, and we're combining the program for 2022. And it's a lot of very interesting liver topics there, non-alcoholic fatty liver disease not being the least of them, and very excited to see what's emerging. And then, of course, happy to discuss today's NASH-TAG agenda in the wake of all of this.
I think it's important to note that cookies and chocolate have more fat content in them than a small bag of Lay's potato chips. Just saying.
I can't comment on that. Of course, you're right. And although when we take into account salt and a couple other things, it's neither is exactly a good ride. But we've now praised your food with faint dams, Stephen. I think is how you put that, right? So here's mine. I'm in Maine with my daughter, and my wife came up with me this time, and that's always great. I love Maine. I love my daughter. I love my wife. All good. Professionally, and the other thing is that the Spurs, who I've been complaining about for a while, finally seem to have gotten themselves somewhat sorted out under Antonio Conte. So I've come to watch matches again without fear, which is a good thing. Professionally, we will be announcing in the next couple of weeks at least 3 sponsorships for this podcast for 2022, and a couple of interesting ideas. As we tossed out a couple of times, what should we do for season 3? And people are starting to come in with ideas. I'll tease one. What would surfing NASH on Instagram look like to you? You'll see, because we're working on it right now, and that should be fun and different. And what does that sound like to you? How do you feel about it? What other ideas do you have? If we're willing to go as far as to consider Instagram, is there a way you can push us into TikTok? Dr. Harrison would like to be our spokesperson on TikTok. I know that for certain.
Come on.
His kids will think that's really impressive.
You should see Michael Charlton move. He can get down with like MC Hammer, big time.
There aren't a lot of Twitter. I've seen so far on liver Twitter, a couple pictures from Laura Kulik, but I would love to see the video of Michael doing Hammer.
Laura Kulik was getting down, no doubt.
These are not get down pictures, though. I got to tell you, these are very, very chill pictures. This is just walking the aisle pictures. Very, very sweet stuff. No boogie anywhere.
There was a lot of boogie happening, but it was after the wedding.
Are we introducing a gossip section now into this?
Stephen, if you want to provide details on that later, we will put it up as part of what's posted only on the website, and that'll give people more reason to go there. I kind of like this. In the meantime, let's go to NASH-TAG. Stephen, your floor.
Sure. So, guys, first of all, NASH-TAG, I think we're in the 5th year counting, right? And it is just blown away our wildest expectations when Vlad and Michael and I sat down and came up with this meeting, and we subsequently brought in Rohit as well. It has become a meeting that people look forward to participating in, look forward to listening to for a lot of different reasons. The fact that it's relatively laid back and people can speak their mind, that it's all focused on NASH with a significant amount of content on drug development and biomarker development. It's steeped in in science. It has made this a very good meeting. And this year, I think, is no exception. So the meeting is January 6th to the 8th at the same place we always have it, the Chateau in Deer Valley. We're going to kick it off on Friday morning, January 7th. That's my birthday, so it's always a good day, with Mike Charlton doing his 2021 year in review. And if you've never heard Mike give his opening address, it literally is 30 minutes of buckle your seatbelt and get a perspective on the NASH landscape that I guarantee you haven't thought about before. Mike, and I mentioned this in his wedding, he is a deep analytical thinker, and he will take something and look at it through the lens that nobody else looks through and come up with some thoughts that are mind-boggling, but that are so true and steeped in experience and wisdom. And I think you're in for a treat this year, just to start the meeting off with Michael and his year in review. And then we're going to mix it up a little bit. Our first session is entitled A One-Stop Shop. And the moderators for this are Rohit Lumba and Maru Ranella. I think she's maintaining Ranella as her last name, even though she married Michael Charlton this past weekend. What we're going to do is a little bit different. You know, before what we did was kind of a pro and a con, where we took a presentation from AASLD or EASL on a new drug that was reporting out, and we would speak to it from a pro and con perspective. Now what we're doing is we're going to take this section and break it down into different mechanisms of action for drugs. So the first one is FXR, and I'm going to present that one on a clinical trial summary of the FXR agonists that are out there. And as we know, not all FXRs are created equal. It used to be we thought they would be the backbone therapy for NASH. I don't think we think that way any longer, at least I personally don't, but I do think they still have a role to play. And we'll talk about what some of those results have shown and how we can accentuate the positive attributes of FXRs for at the same time mitigating some of the class effects of side effects that we see with LDL and pruritus. And there will be a 10-minute open mic discussion, which is another benefit, I think, of the meeting, where we're able to really just, what I like to say is take the gloves off and just have a raw, real session on the data. And then we'll do the same thing with FGF19 and aldafermin. There's been some disparate trial results that have come out from cohort 4 that was published in Gastroenterology, and then the Alpine 2-3 data that was presented at AA. And Vincent Wong will go through that, and that will be another open mic discussion. And then we'll end this particular session with Rohit Lumba speaking on FGF21. Cover to Cover is the name of that, Cover to Cover FGF21 trials. And this is very apropos because I don't know how many conversations I've gotten into since the Peg-Belferman data were presented by Manal and Rohit at AASLD, and a concern arising that maybe the FGF21s are not all that. It's similar to the FXR story in that not all FGF21s are created equal. Some bind FGFR1C beta-klotho, some bind FGFR1, 2, and 3. So you get different amounts of liver fat reduction, potentially different histopathologic impacts, different extrahepatic benefits. So those all are going to be discussed, and then an open mic debate to ensue. That's session 1. I don't know if we want to pause there and before we get into session 2. Maybe I'll just stop there and see if anybody has any thoughts or questions.
Definitely. That's very exciting. You know, if I have to fly there, I'll just fly there for Michael's review of the year 2021. That's a great opener. And it kind of lays the track for then 3 mechanisms of action that are very dedicated to liver pathophysiology in this disease. We're not talking the more metabolically orientated PPAR or GLP-1 here, but these are drugs that affect hepatocytes and the stellate cells and the gut-liver axis, very much focusing on that interaction, fibrogenesis, and disease drivers. So very excited about this first session. I think you guys couldn't have come up with a greater start to the meeting.
Well, thank you. Yeah, I think it's broad-based, but liver-centric in a way. There are opportunities for some good discussion around where do we see these therapies potentially playing out in clinical practice, both as monotherapy and as combination therapy. 2 of these 3 mechanisms are injectables right now. So where does that play a role? So I think it opens itself up nicely for discussion.
Stephen, I agree. And it was a week or 2 ago that I, for the first time, heard somebody use the phrase the FGFs as if they're all the same. Not just the 21s are all the same, but the FGFs are all the same. I was puzzled by the comment, but I will appreciate appreciate seeing these laid out that clearly in this context, because obviously there are people who need to be set straight, or at least to understand a little more than they do right now.
Yeah, I mean, clearly FGF19 and 21, there's a lot more difference between just two numbers, right? FGF19 binds beta-klotho FGFR1C and 4, whereas none of the FGF21s bind FGFR4, which is what modulates CYP7A1 inhibition and conversion of cholesterol to bile acids. So they're absolutely completely different. They do all bind FGFR1C, and that is important in modulating free fatty acid flux from sick adipocytes. That and they're both injectable. So, but I think that's where the similarities end.
Yeah, as I say, I think it would be helpful for people to see that laid out clearly. And then again, in the same context you're talking about, that not all FGF21s are the same. People need to get all that. Okay, good. Louise, comment, thought?
I'm just interested to listen to the data and how we can put that into the real world to support patients, because obviously some of these have different medications that you may need to add in to support side effects. It's more from the practical side that I take from the clinical trials and that aspect, and how we implement it in a patient paradigm.
Well, let me go on and talk about session 2 and 3. At least with session 2, there's some new differences from prior meetings as well. And part of this is we want to mix it up. We want to keep you guys off guard a little bit, and maybe a little bit like a box of chocolates. You never know what you're going to get. So here we turn back to Vincent Wong, and we asked Vincent in session 2 to go over the latest clinical trial results and specific key Congress presentations. That is going to be a little bit like session 1 in the past, where we bring in different key presentations and we have a pro and con. We're not going to do a pro and con, but we're going to have Vincent give a very unbiased view of what he considers to be the presentations that had the biggest impact from a clinical trial perspective in NASH. And then I'm very excited to say we brought back the oral presentations. We didn't have any oral abstract presentations last year. Everything was poster. We have had the best crop of abstract submissions we've ever had at NASH-TAG. Jörn was mentioning he just finished grading the DDW abstracts. I have those in my inbox to do. Jörn always accomplishes tasks much quicker than I do, so I still have that in my do-out box. But I've reviewed abstracts for a long time for all the major congresses, and I can tell you that many of these that were submitted would be taken as orals at AASLD or EASL, and a lot of them are novel. So we have had to scratch and claw this agenda to plug and play quite a few oral presentations. We're going to have one, maybe two of them presented following Vincent Wong's key Congress presentations in session 2. And we're going to smatter them throughout the 2 days of the meeting. We're going to give them 5 minutes to present and 5 minutes for Q&A. So that's session 2. Jumping straight to session 3 is our first shot at going back to biological basis of NASH progression. So we're going to dive back into the basic science part. And here we have Brent Tetry, who is well known in his understanding of the pathophysiology of NASH. He's going to be moderating this with Frank Anania from the FDA, who has also made a career of bench research in the field of NASH. And we're going to have 4 presentations followed by a panel discussion, really kind of cutting edge. So we'll have a discussion on the role of phage therapies in liver disease, then emerging data on lipotoxicity in NASH, and then why is NASH-related HCC potentially resistant to treatment. That's going to be fascinating by an investigator out of Heidelberg in Germany. And then we're going to have a presentation on organoids in NASH therapeutics by Dave Brenner from San Diego. And then we're going to open it up for 20 minutes, and we're going to have a discussion as we go to our lunch break. So that's session 2 and 3. I think it's going to be fascinating. Let me turn it over to my colleagues for comment or questions.
I was going to say that I'm interested in the why NASH-related HCC is resistant to treatment, because it begs that we have to diagnose earlier, we have to start screening earlier, particularly if we have a form of cancer that is really resistant. It really pushes that paradigm further forward for the screening of type 2 diabetes who really suffer from this population.
That's a great point, Louise. I mean, the other things that come to my mind, and again, I don't know what is all going to be presented here, but I think we diagnose rather late in the disease course. It tends to be more multifocal than solitary, and there may be additional factors that come into play in this discussion, but I'm intrigued by what will be presented in this 15-minute talk.
You know, this third section, Stephen, really is linking the basic science back into the program, and this is a great set of speakers right there, and a discussion on what drives the disease, how is it emerging, and we'll learn on ideas on therapies and how we could exploit that as clinician scientists. The data Matthias Heickenwelder's lab lab's been producing is incredible. They published just this March in Nature on the depletion of NASH HCC of immune-activated CD8 cells, or the cancer itself is enriched, but the CD8 cells are exhausted, not capable of launching that immune response that you'd like. And they actually show that the typical HCC drugs we use don't work as well in NASH HCC. And there you go. You can't treat the cancer because it's cancer. You have to consider the underlying disease because NASH has such a specific phenotypic immune activation pattern, and you got to coin that drug you want to use to the patient. And I think this is where this great science really evolves the field. And the same is true for Jörn Schattenberg and his talk on the microbiome. I think we'll learn more from, and he's been doing great stuff all these years. And the topic is actually phage therapies here, and it'll just be very exciting.
For once, I'm not sure I have anything to add at all of any value. So, Stephen, why don't you just go on?
Well, thank you for your comments, Louise and Jörn there. Then we jump straight into this interactive dialogue on NASH topics, and I intentionally call it that because there's a 4-hour break in actual lecture content, but it's anything but a break. I mean, some people use it to go explore the mountain. Others use it to network and to meet with colleagues that they haven't seen in quite some time, or to talk about nuances of NASH, where the field's headed, what their ideas are, how they can link those to other ideas and actually put them in practical use, or even begin to look at exploring various combinations of different therapies. So it really is one of the highlights of the meeting is this 4-hour period where we just take a step back, take a deep breath, and really just get to know our colleagues and have some interactive discussions. What is new this year is we actually have a CME lunch symposium. So from 12:30 to 1:45, we're actually going to have a lecture or a discussion entitled Pearls and Pitfalls, Updates on Emerging Therapies in NASH. And the faculty for this are Michael Charlton and Rohit Lumba, and it is supported by an educational grant from Novo Nordisk and Merck. So collectively, this is a little bit like what you would get at, say, AASLD or EASL, where you would have a satellite symposium. That's what we're doing, or at least the intent of what this meeting is for about an hour and 15 minutes in this afternoon section. And then we'll all come back together at 4 o'clock for session 4 and for the second half of the first day. And this first session is entitled What's Happening in Major Consortia Across the Field. We'll have Arun Sanyal, Quentin Anstey, and Manal Abdulmalik moderating. Again, something new, something novel. We know about these consortia that are really focused on developing biomarkers in NASH. We know of LITMUS from the IMI in Europe. Quentin Anstey is kind of the director of LITMUS. He will be giving an update on where things stand. And then a similar but yet different consortia in the US called NIMBLE is led by Arun Sanyal, and he will be giving the update there. And then we'll bring 2 oral abstract presentations in smack in the middle of this, and we'll follow that up with Vijay Shah from the Mayo Clinic giving us an update on the new NIH-sponsored Cirrhosis Network and where things stand with that. We'll have 20 minutes for question and answers before we go to a break to look at the poster viewing. This, again, is a unique session that provides the opportunity to really understand from these very large and well-organized consortia and networks where things stand today as we roll into 2022, and how close are we to having some impactful data generated from these networks and consortia. And then we'll open it up for discussion to see what else they might have up their sleeve that we can expect coming out of these large organizations as we move through 2022 and into 2023.
Stephen, just a naive question to start. The oral presentations, when will it be announced what those are? And has that been determined yet, or how's that going? How does that happen?
Right now, we have selected 5, but we may wind up with a few more than that because there are just so many awesome abstracts that we want to present orally. It's just trying to squeeze them into the meeting in little nooks and crannies where we can. So far, we have 5, and the authors of those abstracts have been notified. There are a couple where we've got a little limbo action going on because we don't know if they'll be posters or orals. It's still yet to be determined, but hopefully within the next day or two, we'll have that ironed out.
And the other thing is, Stephen, this is really a great platform for those amazing research networks that have been formed on non-invasive biomarkers, as you pointed out. LITMUS presented by Quentin Anstey and NIMBLE by Arun Sanyal, and we saw some of the data at AASLD, Arun presented there. And then, of course, the Emerging Cirrhosis Network. So I think those are investigator-driven or academia-driven consortia where we think the biggest unmet need is, and have really just generated amazing amounts of data. This is really part of the academics bringing in their science here.
Yeah, that's right. And you and I work a little bit more closely with LITMUS, and I can tell you just some of the independent patient data meta-analyses that we're beginning to generate out of that consortia has been quite impactful and is helpful in the NASH field. So we'll see even more data coming out in 2022. And then we still have to get to the end of the story as far as some of the follow-up goes in these biomarkers, and that's yet to come. So maybe we can even get an update from Quentin and Arun on how enrollment's going and some of those key issues, particularly in the middle of the COVID pandemic, kind of how things are going. I think it'll be insightful and instructive, and something that you just don't have at every liver meeting is what's the latest and greatest coming out of these consortia. And then and maybe I'll just go to session 5, which wraps up the first day. This is a discussion on artificial intelligence platforms in NASH trials. And as we know, if we just think back to last year and where we were with AI digital pathology and the huge leaps and strides that have been made just in the past 365 days, it is mind-boggling. And so we have carved out a special session here where Naeem Al-Khoury and Ken Kuzi are going to moderate. We're going to hear from Mazen Nureddin on new data on Path AI and their platform. And then Naeem Al-Khoury is going to talk about new data from HistoIndex. And then Arun Sanyal is going to give a talk on what's next in AI digital pathology platforms in NASH trials. What does the future look like? And then we've carved out a special 15-minute panel discussion where we're going to bring in some of the AI digital pathology key folks. So we'll have Katie Wack, who's the Vice President of Clinical and Regulatory Affairs from Path AI, and Gideon Ho, the co-founder and Chief Commercial Officer for HistoIndex. There on the panel, myself and Quentin will also be there. And then we also have a special guest in the form of Dave Kleiner, a pathologist from the NIH, who will be a part of this discussion. We'll have hepatologists, we'll have pathologists, and we'll have AI digital pathology, commercial and regulatory folks here having a discussion about what's next? Where are we at today? And how can we continue to incorporate this data into what we're doing in NASH drug development?
I just want to say I love this session and session 4 coming right before it. I like the idea of having 4 hours to think through what are we learning in general that's going to make us smarter about what we're finding in studies, right? If AI is about how we get smarter in terms of what we're learning in biopsy, and the consortia are about how to make smarter and more intelligent use of what's not a biopsy, I just feel that we will understand a lot more about how people are going to be looking at things in the year ahead by sitting through this session, and I'm amazingly excited about it.
And you know, there are a lot of contributors. When you step back and look at the amount of data that's pouring into LITMUS and NIMBLE and the amount of brain power that's dedicated to mining it, it really is mind-boggling. Again, Jörn and I are humbled by what we see coming out of LITMUS and the amount of energy that's put into that. And I'm sure the same holds true for NIMBLE and the work Arun and others are doing with that consortium. So the Cirrhosis Network is just getting underway, but it's going to be exciting to hear exactly what's in store there. And again, another area where we are seeing a lot of movement in drug development in the cirrhotic, particularly the well-compensated cirrhotics, and also in biomarker development. And I've said this for a while in the podcast, biomarkers have a much easier path towards recognition and approval through the cirrhotic platform than they do through the non-cirrhotic platform. And it may be the easiest way to allow for broad-based application of an NIT. If we can show it quicker in the cirrhotic population and then take those lessons learned to the non-cirrhotic populations. What I'm hoping to get out of that is kind of where are we at with that, and what are some of the challenges that we still need to kind of work our way through to get us to the end game that we want.
Now, the other thing that came up to me is nice to see a number of solutions for Path AI and histopathology presented next to each other, because they could be complementary, or they could have certain strengths and weaknesses. This is a very fine way to discuss this with Arun wrapping it up and giving an outlook and 2 platforms back to back.
Yeah, I completely agree. And then remember, we also are very hopeful to have FDA representative from the biomarker qualification program as part of this conference as well. And we'll get to that towards the end of the agenda, but hopefully having that person be able to be a part of this discussion as well would be fruitful. Well, let me jump into Saturday. So day 2, how do you top day 1, right? So day 2 starts off bright and early. We get everybody up at the crack of dawn in Utah. So session 6 actually kicks off at 7:30 after a hearty breakfast at 6:30 in the morning. The title of this is What's New Out There and How to Interpret What It All Means. And Vlad Ratsiu, myself, and Brent Tetri will moderate. We're going to hear a lecture from Maru Ranella on an update from the AASLD NASH task force. And I think that is really going to be an insightful lecture that gets at, kind of how AASLD is framing NASH and how we see that AASLD works with other congresses, whether it be EASL, APASL, even cross-specialty with the ADA and some of the other groups that are out there that are focused on diseases that are related to NASH. So we're talking diabetes and obesity, and having an update on where we are with working with our other colleagues in different disease states, but yet in the similar frame of mind. Right after that, Phil Newsom is going to give us new data on wet biomarkers. Then we're going to hear on combination therapy in 2022, what the current status is. And Mike Charlton is going to do that. And then Rohit Lumba will come back with new data on predicting treatment response by imaging. And then there will be a panel discussion following that. This is a data-rich section. So we go from an update from Maru on this NASH task force to, wet biomarkers, to imaging biomarkers, to combination therapy, and then a discussion. There is a section here that is kind of in italics. It's called Lessons Learned from Early Pioneers in NASH Drug Development. That we're going to pivot to the end of the day so that we have more time for discussion around wet and imaging biomarkers as well as combination therapy. That is a rich panel discussion. We have about 20 minutes for that, but this may be one of the most intriguing sessions because it really gets at what we're all wanting to know, and that is, where are we at with combination therapy? Where are we at with these biomarkers? Is there anything we can use in clinical practice today? And how quickly are we expected to have something that could be approved by the FDA or used in clinical practice? And this actually becomes a critical point because we are very close to having our first potential FDA-approved drug. And when you have a drug approved, we need to be able to identify the at-risk population of patients that would be eligible for the prescription medication. And then we need to be able to monitor response to therapy. And ideally, we want to be able to predict outcomes. This is a very apropos session that deals with where we stand relative to that. And then rounding out the discussion with combination therapy, we all know that's where we're headed. Are there some new thoughts surrounding combination therapy or novel ways of looking at it? And I know Mike is going to definitely give us that interpretation as he gives us a very nice lecture on where we're headed. Maybe I'll ask for comments relative to session 6.
Well, so for one thing, I love the idea of Mike starting by talking about where we've been and then being the guy who shows up at the end of this and talks about where we're headed, because knowing Mike, I have a sneaking suspicion that there will be themes that will run through those 2 talks that will not be things anybody's thought of before they get in the building.
Absolutely. And they may or may not be thoughts that are not only appreciated, but they may or may not be thoughts that people might have a visceral response to. That is one of the beauties of NASH-TAG. You take the gloves off and we have a raw discussion around what works and what doesn't work and why we should be putting things together and why we shouldn't. Mike does an amazing job at balancing that very, very well.
I think it excites me because I enjoy looking at the treatment responses by imaging. I think that's where obviously we're headed. We're trying to get beyond the biopsy as much as possible. I think obviously the wet biomarkers looks like it's going to be a great session. The problem for that with me is we've got to get the football through the door to get the wet biomarkers taken, and the majority of people remain outside of mainstream healthcare. The imaging session again adds to, can we pick these people up who should be then going for wet biomarkers? So it becomes that circular argument of picking people up through imaging that we can say are higher risks and then putting them through the wet biomarkers to stratify and look for combination therapies. So it looks like a very good session to me.
Well, and this one part that we're moving is this lesson learned from early pioneers in NASH drug development. When we were talking about the agenda for this meeting, one of the discussions that came up were, there's a lot of drugs that have been put through this drug development process, and many haven't made it. There are a lot of people that have been a part of those quote unquote failures, but there are tremendous lessons that could be learned from those failures. I mean, Michael Jordan said it best, I've learned more from my failures than my successes. So being able to have some of these pioneers come back and say, hey guys, hashtag real talk, this is what I learned from doing this. We don't want to make those same mistakes over and over again is a fabulous section. But to give it just due, we moved it out of this particular session and to the last session.
If you mentioned it now, I'll mention it now too. You know, I'm mindful of the idea that if every episode that we did in the first year of this podcast, the best received and most listened to episode that was not a news event or part of a conference was Jörn's talk about the paper about why drugs fail. So I think there's immense interest in what people can do to learn from those mistakes. I saw it in the example I just gave. I've seen it in other places. So the idea that you're doing this here, particularly in a moment when folks have a lot to be excited about, is a little bit like, okay, let's pump the brakes for a minute. Let's make sure that we've learned all the lessons that we need to so that we don't replicate mistakes. I think that's another thing I'm really excited to hear about when it comes up.
All right, well, let me keep rolling here. Now we're at session 7, starts at 10 o'clock in the morning, and this is titled Novel Targets. Naga Chalasani, Celeste Thomas, and Phil Newsom are going to moderate this session. We're going to get a talk from Luca Valente on PNPLA3 knockdown as a treatment of NASH, and that's going to be followed immediately by Jörn Schattenberg on HSD17B13 knockdown and inhibition as a treatment of NASH. Two very prominent single nucleotide polymorphisms that are now being targeted for drug development. We'll hear the latest and greatest on that. We're then going to pivot to a talk on MOTC in the treatment of NASH, and then uncouplers in the treatment of NASH by probably one of the most famous endocrinology researchers on the planet, and Gerald Shulman from Yale, looking at where we are with some of these novel treatments in the field of diabetes. Actually, a couple more wonderful lectures targeting the gut-liver axis in MAFLD using CarbAlive. This is an interesting talk by Rajiv Jalan from the University of London. And then mechanisms of VLDL turnover implications in NASH therapeutics by Dave Cohen from Harvard Medical School. And then a very nice panel discussion for 30 minutes that kind of covers all of these novel therapeutic opportunities.
I like this one because when you were talking about your interactive dialogue on NASH topics from Friday, I presented Jörn Rotman's stuff on genomics. Probably not as well as he did, but I did a summary for our thoughts. Cast, and in fact, he should have a speed date with the HCC resistant to treatment, because the one part of his genomics session and the summary at the end was it was very good at targeting people with and detecting the HCC groups. So that precision medicine. So if I'm going to make a speed date, it would be those two. I was quite excited to see him on the agenda, particularly on his favourite topic, which is this particular polymorphisms. And he just presented very well on both of them at AASLD.
Well, just to get through this in time here, we then have a 4-hour break for an a la carte roundtable discussion related to drug development. Again, your options to do what you'd like to do. We do, again, novel to this year, have another CME lunch symposium entitled New Advances in NASH Diagnostics and Prognostics. And there we'll hear from Mazen Nureddin. And then we jump right into coming back from that break. At 4 o'clock, we have a couple more oral presentations, and we get into some basic science. We talk about single cell transcriptomics in NASH, and then inflammation and gut dysbiosis, and then integrins and fibrosis, as well as an overview of dual and triple agonist in the treatment of obesity by Sam Klein. Any thoughts on this section before I roll to the last session?
Stephen, looking again at the program, it interacts so nicely with the clinical data and then again the basic science, educating us on state-of-the-art data that's been pouring out of these great labs. You know, the single-cell transcriptomics, we've discussed this here with Scott Friedman and the implications for the field. And then Neil Henderson gave great talks at ASLD, same topic, and mixing this with the clinical trial results. I think this is really what makes NASH-TAG so special.
I agree. Yeah, thank you. We top it all off at the end of the day with what I am entitling a fireside chat. Actually, 2 of them. Literally, this will be a fireside chat. NASH endpoints and regulatory landscape. Myself and the co-directors broke this into 2 sessions. Non-cirrhotic trial endpoints. Is it time to pivot? I'm going to present just where we stand with the data today, and then we're going to open it up for a long discussion, because I really think this is the crux of the matter in drug development for NASH right now, is endpoints and where we stand understand, and can we get alignment on moving beyond the biopsy? If we can't, how can we refine what we do today to make it easier to not only enroll patients, but to get to an achievable endpoint without lots of noise that we hear and see with the variability that exists today? And we have both industry representatives, regulatory representatives, key opinion leaders, all at the table having this discussion, fabulous discussion. And then immediately following that, Vlad is going to stand up and give us the latest and greatest on cirrhotic trial endpoints. His title is, Are We Ready for NITs? And very brief 15-minute talk, and then we'll open that up to another nice discussion, fireside chat, again, with the same group of participants. This may be the highlight of the session, kind of wrapping up all this at the end. Everything we've talked about for 2 days are kind of brought to bear in these 2 fireside chats. And then I'll wrap up the meeting with a recap and look to the future. Ultimately, this has promised to be the best NASH-TAG yet, and I'm super excited to be able to talk about that today with you guys.
Stephen, I'm thinking the last session has the promise to be the best session anybody's ever seen anywhere in fatty liver disease, if in fact it becomes the kind of exchange it's capable of being. I just find it so exceptional you guys have pulled that together. It's just going to be such a remarkable event to watch.
Thank you. I think it's a testament to the fact that every one of these stakeholders feels the exact same way, from the FDA to sponsors to key opinion leaders and everybody in between. Everybody wants to find a way to get the right drugs to our patients as quickly as we possibly can and as safely as we can. And so we all have the end goal the same, but how do we bring it all together and kind of hit the accelerator a bit to get us where we want to be?
Is it your sense going into that, Stephen, that not only does everybody have a common frustration, but common specific pain points around that? Because the easiest way to move something forward, right, is to figure out where the common pain points are?
I do. I think people look at it through a different lens depending on which seat they're in. And the common theme is patient first, safety first. I know that's the lens that the FDA looks at this through. From the investigator perspective, or maybe my perspective and Jörn's perspective, I don't want to put words in Jörn's mouth, but we look at it from a clinical side and we see the urgency for which we need to get something through, but we also see that there are some challenges with the way that we're currently looking at it. It's not inherently a pathology problem. It's not inherently a regulatory problem. It's understanding the nuances that the liver is heterogeneous, that this liver disease is heterogeneous, that the patients are heterogeneous. What do we need to do to identify, if we're moving beyond the biopsy, what data do we need to generate to make everybody feel comfortable that we can take that leap? And if we're not ready to take that leap, maybe how can we adjust, modify our histopathology endpoint? Maybe it's including AI digital path. How do we put that intricate puzzle together in such a way that ultimately we can get a drug or multiple drugs approved to treat this disease as quickly as we can? And I think that's the hope and goal and thrust of these 2 fireside chats.
Stephen, I'm going to let that be the last word because it's so dead on and so optimistic. And in the moments we've got left, let's just go to final question. My final question today is for each of you. Is there one thing in this agenda you see that we haven't talked about for more than, say, 10 seconds in a row on this podcast that you'd like to point out because you think it's going to be interesting or worth people looking at?
I think it's a great program. And, at the point of time we are, no drug approved. We're struggling with some of the endpoints. What do we want to measure as a primary endpoint? This is exactly the right program. Moving forward, there will be some room for how do I implement this? I need a patient in there on the fireside chat to discuss what does the patient want. But we don't have that drug yet. We can start thinking about it, but for sure what the field needs is what Stephen and colleagues really assembled here to define. This is the endpoint. This is the mechanism of action. This is my biomarker. And here we go.
I was just going to echo exactly what Jörn said. It's an absolutely fantastic program. And I know it's a science-based meeting, but the one for me is the missing the patient, particularly around the fireside chat. I can think of a few patient advocates who would be comfortable with a fireside chat. That was the only thing that was missing. But other than that, I think the agenda is absolutely fantastic. I'm excited to listen. It might be an early morning fireside sleep for me, but...
Louise, since I suspect we all know the patient advocate you're talking about, I know she tends to be in the audience. I would be surprised if she were silent throughout the whole conversation just because we know and love her. I'm in the same place that you guys are, and the forecaster in me takes a look at one thing in this and says, gee, that's interesting, which is the Valenti and Rotman talk on the knockdowns of PNPLA3 and HSD17B13, particularly HSD17B13. 2013, as Michael Charlton, who owns my two favorite out-of-the-way comments of the year, said when talking about AASLD, if we're looking for where a vaccine might come from, he talked about, if you recall, the Arrowhead late-breaking poster on HSD17B13 variations. Shortly after that, Novo Nordisk goes down and plunks down $3.9 billion on Dicerna, and everything Novo Nordisk does really is somewhere over in this metabolic space. So it might be on type 1 diabetes patients, which is an area of interest of theirs right now, or it might be things more about NASH. But I'll be interested to see if there's anything we learn in these talks that start to forecast out the future of those agents and the potential for vaccine. Because if when we get there, that will be a really exciting moment for the patient population and the commercial community. And this is the first thing I've ever seen on a program, short of the poster that Michael pointed out, that says, well, gee, we may be ready to start talking about that. So I'm hopeful. Stephen, what's your favorite moment besides all of them?
Well, that's like asking a parent who's their favorite child is. I'm a little prejudiced to the fireside chats. That's where we have the right people at the right seat to have a frank discussion and try to build consensus around where we need to go. To Jörn and Louise's point, it would be great to have a patient there as well, but we don't have that this year. But I think it's still going to be an incredibly helpful session and hopefully something that we can take away from. And, that's one of the things we always want to take away from these conferences is what can I take back and use in my clinical practice? Well, this isn't a clinical practice meeting. This is a research meeting. What can I take out of this meeting that changes the way we do clinical research moving forward? If we can do that, this will be the best NASH-TAG ever.
Okay, I'm going to let you have the last word twice because that's a perfect closing statement for this episode. So thank you with that. Listeners, I'm going to come back into business section in a couple of minutes. Stephen, Jörn, Louise, thanks for getting this done at a slightly different hour for us. And Stephen, thank you for laying it out so neatly. What a great presentation and what a great meeting. I'm excited.
Well, thanks, guys.
So we will see you all next week, and I'll see you for business section in a couple of minutes, and we'll see you guys soon, okay? Thanks. Bye-bye now.
Welcome to today's episode 60 business section.
Hi, listeners and downloaders. Are you ready for NASH-TAG? After that session, I certainly am. For those who want to register for live or virtual conference, I believe that going to the NASH-TAG 2022 website will point you to the place to register. If you can get to Deer Valley, you really should go. As we've been saying, there's nothing remotely like it on the NASH-NAFLD calendar, particularly if you're there live.
We'll be covering NASH-TAG daily from the Chateau in Deer Valley, Utah.
The current plan is to record our sessions before the day starts on Saturday and upon waking up on Sunday. Remember, the session closes Saturday night, so Sunday might be a little bit later. Our panelists will be conference attendees, and we'll have live audience available for anyone awake at that hour, which will be easy in Europe, reasonably easy in the eastern U.S., a bit of a challenge in Chicago, and really tough for the Mountain and Pacific time zones. Keep your eyes out next week for more news on our panelists and our recording times.
Apologies to those who tried to get into Monday's live audience.
We had to change the recording time twice in the 48 hours before the session to align with some issues for two of our panelists. If you came to the audience at 3 or 3:15 Eastern Time Monday, U.S., and nothing happened, we were done by then. This coming Monday, we'll be back to our regular time. Please accept our apologies.
What's coming in the rest of December?
Next week will be our last session of the year, recorded during that same week. Lead author Ali Amirian of the Cleveland Clinic and co-author Naeem Alkhouri will join the regulars to discuss the groundbreaking Splendor study. This should be fascinating. And then the last 2 weeks of December, we'll have individual interviews with some of the people who made 2021 such an exciting year for Nashville, NAFLD World, and this podcast. Again, keep your eyes open for more information and listen to next week's business section.
This week, the Buzz and Vault section is pretty low-key.
First, the buzz. This week's number is 377%. That's the increase in average daily listening downloading for the past 4 months compared to the same period in 2020. Not too shabby. This week's Vault report starts once again with the Paris NASH imaging conversation 46.2, which gained another 90-something new downloads last week to go over 750 as of 5:30 p.m. Eastern Time on Tuesday. The 57.1 from the AASLD wrap-up episode is still outpacing that total episode, and people are increasingly listening to the conversation more than the episode, which tells me there's something good there you might want to listen to still. Nothing in year 1 or early year 2 stood out this week, but earlier this year, episode 42, our discussion about FGF with Arun Sanyal continues to grow. The most interesting story this week, though, is that the 2 same-day AASLD episodes, 55 and 56, continue to grow rapidly. Both have broken into our top 10 all-time episodes, and day 3 is about to become the second episode in the past 6 months to break into the top 5 after the renowned 46.2 Paris NASH conversation. And with that, I want to thank today's superb panelists, our engineer Mike Wilson, who is still saving Louise's voice or sound on a week-by-week basis. We're working on it. Eric Rounds, who's been driving social media, Steve Ennin, who helps make this whole thing make sense. Finally, most importantly, it starts with you, listeners, who provide us the energy and the inspiration to do this every week, 50 weeks a year. Not the last 2 weeks in December, at least not live, but you'll hear about that next week. We'll be back next week to discuss Splendor, which is an amazing study. Until then, stay safe, surf on. We'll see you next week on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.